Would you like to learn about cell biology?
Sure, sounds awesome.
Each and every cell on this planet is covered in a plasma membrane that separates the cell from the outside environment. Membranes are gatekeepers of the cell since they allow only certain molecules to enter or leave. Cell membranes are a portal and a barrier. Lipids provide the barrier and proteins are the portals. Membrane proteins are pumps, channels, and receptor proteins. Pumps use energy to move molecules from areas of low concentration to areas of higher concentration. Channels work the opposite. Molecules can pass through channels as their flow takes them. Receptor proteins can sense, detect, and respond to signals that are sent between two cells or sent throughout the entire organism.
Is there another name for the cell membrane?
Yes, it is also called the lipid bilayer.
Why is it called the lipid bilayer?
Because it is made up of two opposite-facing layers of lipids. The lipid bilayer makes a barrier between the inside of the cell and the outside by blocking the diffusion of most hydrophilic molecules. Lipids are water-insoluble. They have a hydrophobic tail of hydrocarbons and a hydrophilic head containing charged groups. Having this tail and head makes them amphipathic. When amphipathic molecules are dropped into water, they huddle together to keep their hydrophobic tails away from water with their heads facing out towards the watery environment. This results in a micelle being made. Membrane lipids have two hydrophobic tails, one with a kink in it, and are too bulky for the center of a micelle, so instead they pack together tail to tail. The hydrophilic heads face the watery environment both inside and outside and create a hollow ball. This hollow ball is called a liposome.
Wow, that's awesome. How are membranes self-sealing?
I'm glad you asked. When a hole is created in a membrane, the tail groups become exposed. The tails do not like being exposed, so the membrane will spontaneously fold onto itself, closing the exposed area.
Wow, awesome.
The lipid bilayer is also a two-dimensional fluid. The membrane lipids and proteins float in their environment just like icebergs do in the ocean. But they can only float front to back or side to side, never up and down. Floating like that is called lateral diffusion, and that is why it is a 2D fluid. Membrane components cannot easily flip upside down. Membrane lipids will flip upside down roughly once a month. Membrane lipids flipping upside down is called transverse diffusion, and enzymes called flipases exist solely to reverse transverse diffusion and put the lipid back in its correct position.
Interesting.
The fluidity of a membrane is based on how closely membrane lipids can pack together. The more tight and denser the lipids pack, the less fluid the membrane is. The membrane is reinforced by a meshwork of proteins called the cell cortex that lies just underneath it. The exterior face of the lipid bilayer, the face that is facing outward, is made up of glycolipids. These lipids have sugars attached to their heads. These sugars are called oligosaccharides. Many membrane proteins also have sugar complexes attached to their exterior faces. They are called glycoproteins. All of this forms a sugar layer on the outermost exterior of the cell called the carbohydrate layer. The carbohydrate layer protects the cell against environmental damage. The sugars can also absorb water, which makes the outer edge of the cell gelatinous and lubricated, which helps motile cells squeezing into tight spaces. The lipid bilayer is asymmetrical, meaning it has two different and distinct faces. The lack of transverse diffusion actually conserves the asymmetry, because if too many lipids were flipping and flopping, then the two faces would eventually become homogeneous.
Wow, that would not be good. What are the membrane proteins?
Membrane proteins carry out most of the activity and functions of the cell membrane. Their main function is transport. If a membrane protein extends through the entire membrane, they are transmembrane proteins. Integral proteins are embedded in the membrane, while peripheral proteins are covalently linked to lipids and are not embedded in the membrane. Since the interior of the lipid bilayer is hydrophobic, almost all hydrophilic molecules are blocked from diffusing across it. In order to bypass this, there are integral, transmembrane transport proteins. A transporter is a protein that contains moving parts and can transfer molecules from one side of the membrane to the other. They usually transfer molecules against their concentration gradient, so they require energy. Channels are also proteins, but they have no moving parts. They create small hydrophilic holes in the membrane which can only be used by a select few molecules. Most channels are ion channels and are specifically for small inorganic ions. Channels require no energy because the molecules are following their own concentration gradient. Cells need to maintain ion concentrations inside themselves since they differ a lot from the ion concentrations outside. Sodium ions are the most plentiful ion outside of the cell and potassium ions are the most plentiful inside the cell. If the electrical charge balance were skewed, the membrane could be torn to pieces. Small nonpolar molecules can easily diffuse across the cell membrane all by themselves. Uncharged polar molecules can also diffuse if they are small, but if they get bigger, they can't diffuse as easily and require transporters or channels. All ions and charged molecules are virtually blocked from any diffusion no matter the size. Channels allow molecules through that have the right size and charge. Transporters, on the other hand, are very specific. The molecule that is being transported has to fit perfectly into the binding site like a lock and key. Once the molecule binds, the transporter changes shape and forces the molecule across the bilayer. Channels and some transporters are responsible for facilitated diffusion, which allows molecules to follow their concentration gradients. Specialized transports called pumps move molecules against their concentration gradient. This kind of movement is called active transport and requires ATP. Charged molecules have the push or pull of their concentration gradient along with the push or pull of their electrical charge. These two forces, one concentration dependent and one charge dependent, affecting a molecule's movement is called the electrochemical gradient. When both forces are in the same direction, there is a strong force pushing the molecule across the enzyme. If the two forces oppose each other, it's a tug of war, and whichever force is stronger pushes the enzyme to the opposite side. It is even possible for both forces to be opposite and equal, resulting in zero movement of the molecule.
How does active transport work?
There are three main strategies for achieving active transport across an opposing electrochemical gradient, and they all require energy. The first is coupled transport. Coupled transporters use one molecule moving with its gradient to power the movement of another against its gradient. The second is ATP-driven pumps that use energy obtained from the hydrolysis of ATP to force the movement of molecules against gradients. The third is light-driven pumps that use a photon of light to force a shape change that moves molecules against its gradient. The sodium-potassium pump is a pump in an enzyme. It is an ATPase and hydrolyzes ATP. It pumps sodium out of the cell against its gradient while allowing potassium into the cell. The sodium-potassium pump helps maintain osmotic balance. Water follows ion concentrations, so as sodium levels climb in a cell, water rushes in after it. The water diffuses through the membrane and flows in through aquaporins. All of this water flowing in puts the cell at risk of bursting, so by pumping sodium back out again, it helps to keep water from staying in the cell. Ions want to move to the outside of the cell since it is negatively charged. They can only move if ion channels are open, so when it's not, it creates a membrane potential.
Very interesting. What's next?
Next we will discuss clathrine-coated vesicles. Transport throughout the entire endomembrane system is carried out by a continual process of budding and fusion involving transport vesicles. Transport vesicles carry soluble, or hydrophilic, proteins. Vesicle recognition and routing depends on proteins embedded in the vesicle membrane. Certain vesicles go to specific places carrying specific cargo. Most vesicles are covered in a unique protein coat. They are called coated vesicles. The coat helps to form the bud, hold it in its shape, target the vesicle, and ensure it has the proper contents. Before fusing with its target, the vesicle sheds the protein coat. Doing this allows the membrane of the target to fuse with the membrane of the vesicle, which then leads to the cargo being released. The best studied vesicle coats are made of clathrine. Clathrine-coated vesicles bud off of the Golgi for secretion and bud off of the cell membrane for endocytosis. At the cell membrane, each vesicle starts off as a clathrine-coated pit, which is a basket-like structure with clathrine protein mesh as its base. Dynamin, a small protein forms a ring around the neck of the vesicle to pinch it off of the rest of the membrane. Once the neck is constricted to the point of breaking, the vesicle is released. A second class of vesicle coat proteins called adoptines secure the clathrine cage to the vesicle membrane. They also choose a cargo that will be loaded into the vesicle. Proteins destined for other targets have transport signals on them, which are a specific sequence of amino acids. The transport signals bind to cargo receptors. Adoptines help capture specific cargo molecules by trapping receptors below the neck of budding vesicles. Once a vesicle buds and is released from its point of origin, it is to find its way to the correct destination, shed its protein coat and fuse with that membrane and release its contents.
How does that happen?
Once the vesicle reaches its target organelle, it is to recognize the membrane and the membrane has to recognize it. Once both are recognized, the vesicle docks with the membrane. Rab proteins on the vesicle tell the point of origin and the cargo it is carrying. Rab proteins are recognized and bound by tethering proteins present on the target membrane. Tethering proteins are specific and unique to only bind to Rabs on vesicles that come from places that should be dumping their cargo here. Once Rabs and tethering proteins have interacted, interactions between vesicles snare proteins or V snares and T snares on the target membrane stabilize the docking process. Now the membranes have to fuse so the vesicle can release its contents. The fusion results in the vesicle membrane becoming target membrane temporarily. The snares will twist, winching the vesicle in close enough for fusion to happen. Now the vesicle has docked and fused.
That is very cool.
Now we are going to discuss the 3 types of cytoskeleton fibers.
Yeah, I'm so excited. The cytoskeleton fibers are my favorite part.
First, you need to know that the cytoskeleton mediates shape change, organization of the location of their inner components and interactions with their environment. Cytoskeletons are the bones and muscles of the cell. Without them there would be no cellular mobility and motility. The cytoskeleton is made up of 3 different types of protein filaments: intermediate filaments, microtubules and actin filaments. The intermediate filament's specialty is tensile strength. They help the cell withstand mechanical stress. They are called intermediate because their diameter is between microfilaments and microtubules. Intermediate filaments are made like ropes. Individual fibers twist together into strands, which then twist together into rope. Pairs of intermediate subunit proteins wrap around each other to form dimers. 2 dimers come together to form a staggered tetramer, and then tetramers bundle to form the rope-like structure. The 4 main classes of intermediate filaments are keratin, vimentin, neurofilaments and nuclear lamines. Lamines are dynamic because they disassemble and reassemble with each round of cell division. This disassembly and reassembly is controlled by phosphorylation by kinases and dephosphorylation by phosphatases. Microtubules play a crucial role in all eukaryotic cells. Microtubules grow out of and originate from the centrosome. They then extend out from the centrosome to the interior face of the cell membrane, creating protein tracks throughout the cell. Walking proteins, also known as motor proteins, walk on these tracks to move cargo around the cell and to transport vesicles. Microtubules also anchor organelles to keep them from floating away. For mitosis, all of the cytoplasmic microtubules disassemble and then reassemble as the mitotic spindle. Microtubules are made up of a subunit protein called tubulin. Tubulin proteins are dimers of 2 globular proteins. The dimers stack together, creating long chains of globular spheres that alternate beta alpha and so on. The long chains are protofilaments, and it takes 13 of them interacting side by side to make up a single hollow microtubule. The alpha subunit is the beginning and the beta subunit is the end. This means that microtubules have polarity. This polarity is crucial to their function. The centrosome is the tubulin organizer. It coaxes tubulin to form the first microtubule tube, and then more tubulin easily joins. The starting point for microtubule formation is a ring-shaped structure called the nucleation site. The negative end of the new microtubule is embedded in the centrosome, while the positive end points outwards. Microtubules will continue to grow until it makes contact with another structure. Then suddenly the entire microtubule disintegrates as quickly as it forms. The constantly changing assembly and reassembly is called dynamic instability. To stop microtubules from falling apart and give them stability, they need to be linked onto something on both the positive and negative end. The cell does not have any way to target a microtubule to a specific location, so the centrosome is just constantly sending out microtubules in all directions at all times. Some will catch on a target and become stable, and others are retracted. This process is very much like fishing. This strategy allows the cells to set up their highly organized and interconnecting system of protein fibers. Actin is found in all eukaryotic cells and is essential for cellular movement. Each actin filament is a twisted chain of identical globular actin subunits which all have polarity pointing the same direction. Just like microtubules, they have a positive end and a negative end. Actin filaments are thinner, shorter and more flexible than microtubules. They are also not hollow. Actin filaments can grow on either end, but growth is easier and faster on the positive end. Each free actin monomer is bound to a GP. Once a GP is cleaved to a DP, the strength of binding for the monomer in the filament is reduced and it will depolymerize. Actin filaments are dynamic, so that they can easily assemble and disassemble. The concentration of free monomers is quite high in the cell. Small proteins in the cytosol bind to the actin monomers and block them from joining growing actin filaments. These small proteins regulate actin filament polymerization and keep actin monomers free and in reserve, so when new actin is needed it can easily be assembled. Most actin binding proteins bind to already polymerized filaments and stabilize them. The cortex previously mentioned that lies just under the cell membrane is actin-rich. The actin provides mechanical strength to the membrane. The actin in the cell cortex also dictates the shape of the cell and how dynamic the cell is. Cell crawling depends on actin. Cells sense environmental signals and migrate towards them using actin. This is a three-step process. First, the cell pushes out protrusions, also known as podia, at its leading edge. In this step, thin sheet-like lamellipodia are pushed out towards a signal. These contain a dense meshwork of actin that is oriented so that most of the filaments have their plus end close to the plasma membrane. Smaller protrusions called filopodia are also pushed out towards a signal. Filopodia contain around 10 to 20 actin filaments. Actin polymerizing into the lamellipodia and filopodia is what pushes the cell membrane out. The second step is anchoring. When the lamellipodia and filopodia touch down on a crawlable surface, they need to stick to, but the anchoring needs to be reversible so that the cell can let go and keep moving forward. Integrins allow for anchoring. They adhere to molecules that the cell is crawling on the extracellular side. Integrins are anchored to the inside of the cell by being linked to actin filaments. The third step is the rest of the cell flowing forward to the anchoring points using contractile forces between actin and myosin. Then the process just keeps repeating. Rho is a small G protein that when activated can trigger actin polymerization and bundling for filopodia, trigger lamellipodia formation and trigger actin-myosin interactions to promote cell crawling. Rho is signaled to take on GTP when receptors bind to the appropriate signaling molecule signaling the cell to crawl.
Wow, the cytoskeleton components really are amazing. What's next?
Next we discuss G protein coupled receptors. First, you need to know what cell surface receptors do. Most signal molecules cannot enter the cell via diffusion, so they have to leave their message at the door. They bind to cell surface receptors present on the exterior face of the cell membrane. The receptor binds to the signal molecule and changes its shape. The shape change transmits the signal into the cell. The signal/receptor complex is the primary messenger. Signal transduction is when the message is passed from one intracellular signaling molecule to another. The other molecules that the message is passed to are the second messengers. These second messengers trigger each other like dominoes until the cell responds. This is called a signaling cascade. One of the categories of cell surface receptors are G-protein coupled receptors. G-protein coupled receptors are the largest family of cell surface receptor proteins. G-protein coupled receptors carry the signal into the cell by a series of modified proteins, usually as a cascade and usually by phosphorylation. They are transmembrane proteins found at the cell surface.
How did they get there?
I'm glad you asked. They were synthesized at the RAF-ER and threaded through the ER membrane using start and stop transfer sequences. They were then shipped to the Golgi, processed and shipped to the cell membrane via transport vesicles. G-protein coupled receptors are members of the 7-pass transmembrane receptor protein superfamily.
So how do G-protein coupled receptors work?
Well, first a signal molecule binds to it, causing it to change its shape. The change in shape by the G-protein coupled receptors activates a G-protein that is associated with the receptor at the cytosol face of the membrane. This means that the G-protein changes its shape and decreases its affinity for GDP and increases its affinity for GTP, so the G-protein binds to GTP, becoming active. Once activated, the alpha subunit can alter target molecules. This initiates a signaling cascade that flows into the cell. Once the signal is no longer needed, the G-protein GTP into GDP and turns itself off. Cyclic AMP is a secondary messenger that gets the message into the cell. It has no informational value, just the presence of cyclic AMP is what conveys the message. The cell itself is programmed so that presence of cyclic AMP means some predetermined message. So signal molecules bind to G-protein coupled receptors, which then activate G-proteins, which activates adenylate cyclase, which activates cyclic AMP, which then activates protein kinase. A protein kinase is what achieves the physiological response. Cyclic AMP is a secondary messenger that gets the message into the cell. It has no informational value, just the presence of cyclic AMP is what conveys the message. The cell itself is programmed so that presence of cyclic AMP means some predetermined message. So signal molecules bind to G-protein coupled receptors, which then activate G-proteins, which activates adenylate cyclase, which activates cyclic AMP, which then activates protein kinase. A protein kinase is what achieves the physiological response.
Very cool.
Next we will discuss the cell cycle.
So exciting.
The cell cycle is divided into 4 phases: M phase, G1 phase, S phase and G2 phase. The last 3 together are referred to as interphase. Mitosis and cytokinesis make up M phase of the cell cycle. G1 phase is the gap between M and S phase and is for preparing for DNA replication. S phase is where DNA is replicated. G2 is the gap between S phase and M phase and is for preparing for mitosis. The cell cycle control system is a network of proteins that monitor and signal the readiness of all the cellular components for division. The cell cycle control system oversees the checkpoints. There are 3 main checkpoints, and if there is anything wrong at any of the checkpoints, the cell cycle will arrest. The first checkpoint is in G1 and confirms that the cellular environment favors division. The second one is in G2 and confirms the completion of DNA replication and repair. This checkpoint is sensitive to DNA damage. The last checkpoint is in M phase and occurs in anaphase. This one confirms that the mitotic spindles are attached to sister chromatides. The most important checkpoint is the G1 checkpoint. Cells require good nutrition to allow cell growth and proper coordination with other cells. Without these, the cells shouldn't divide. When this happens, the cell will pause in G1 indefinitely and can drop into a cell cycle silent state called G0. In G0 they still do all of the normal things, they just don't divide. The cell cycle control depends on cyclically activated kinases, or CDKs. Proteins are regulated through phosphorylation and dephosphorylation. Kinases phosphorylate and phosphatases dephosphorylate. A set of proteins called cyclins control the kinases. The kinases responsible for the cell cycle are CDKs. An example of this is in mitosis when M-cyclin binds to and turns on its mCDK. When M-cyclin levels are high, mCDK turns on all mitosis-needed proteins so that mitosis can occur, and when M-cyclin concentration drops, the mCDK can no longer be active and all of the mitosis proteins are turned off. Each different phase of the cell cycle has their own type of cyclin. Each cyclin CDK complex will trigger different cellular events to prepare for the next cell cycle stage.
How does the cell cycle control system delay its progression to the next phase of the cell cycle when the previous phase is not complete?
This is partly achieved by CDK inhibitor proteins that directly bind to CDK and inhibit their kinase activity. Doing this pauses cells at a checkpoint. If the intracellular and/or extracellular signals favor cell cycle progression, the inhibitor protein is removed from the CDK, allowing it to be active again. If the signals do not favor it, it will arrest. DNA damage causes an increase in the expression and activity of p53. p53 directly binds to DNA and activates the expression of a CDK inhibitor p21. p21 binds to G1/SCDK, and SCDK prevents cells from moving into S phase. This arrest allows the cell time to complete DNA repair. If the damage cannot be repaired, p53 causes the cell to undergo apoptosis. p53 is a tumor suppressor protein that is encoded by a tumor suppressor gene. Once the cell passes through these checkpoints, it has replicated its genome and progressed through G2 and is now ready for M phase. Everything that happens in M phase is controlled by mCDK, as discussed earlier. So now the cell needs to prepare for chromosome segregation. The centrosome has to be copied so that each copy can move to opposite cell poles. Then the centrosome organizes microtubules into the mitotic spindles. When the duplicated centrosomes separate they from esters. The formation of the mitotic spindle begins in prophase. At the beginning of mitosis, the dynamic instability of microtubules increases dramatically and a greater number of microtubules are sent out from the centrosome. Some microtubules will touch microtubules growing out of the other centrosome, and this stabilizes both of the microtubules and forming the mitotic spindle. Now the mitotic spindles are in place and the chromosomes are replication. All that's left is attaching sister chromatids to the spindles so that they can be pulled apart. At this point, the cell is in prometaphase. Here the nuclear envelope will break down into many scattered vesicles of nuclear membrane. The chromosomes are now out of the nucleus and in the cytoplasm. Now the spindle microtubules attach to sister chromatids directly through protein complexes called kinetochores. Sister chromatids are linked together by coenzyme protein rings, and in order to separate sister chromatids we need to get rid of the coenzyme rings. The microtubules attached to the sister chromatids creates tension. The chromosomes are pulled back and forth until they end up lined up at the metaphasic plate. This is metaphase. The tension from the microtubules on the kinetochores of sister chromatids signals to the cell that all sister chromatids are attached and are ready to be pulled apart. The M phase checkpoint monitors the tension, and if the tension is not present, M phase will arrest. But there is tension, and so the cell has completed metaphase. Now the sister chromatids need to be separated and pulled to opposite sides of the cell. This occurs in anaphase. The beginning on anaphase is marked by the release of the coenzyme linkages that keep the sister chromatids together. Now the sister chromatids can be pulled apart. The sister chromatids being pulled apart is called segregation. Separase chews up the coenzyme proteins in order to separate the sister chromatids. Anaphase promoting complex activates separase and targets mCDK for destruction. The sister chromatids being pulled apart is actually two separate processes happening at once. In anaphase are the microtubules that are attached to the kinetochore are shortened by depolymerization, dragging the chromosomes to each pole. In anaphase B, the spindle poles move further apart from each other, and this also pulls the chromosomes further. Anaphase is driven by motor proteins using energy from ATP hydrolysis to disassemble tubulin subunits from the positive end of the spindle microtubules at the kinetochore. Now that the microtubules have been disassembled, we can move into the final stage of mitosis, telophase. The vesicles of nuclear membrane that we made in prophase cluster around the segregated chromosomes, eventually fusing and reforming the nuclei. The nuclear envelope and lamina reforms, the pores pump in the nuclear proteins, the chromosomes decondense and gene expression resumes. Now cytokinesis splits the cell and marks the end of M phase and a complete cell cycle. The contractile ring made of actin and myosin is what contracts and pinches the cell in half. Now mitosis is complete and we have 2 new daughter cells.
Wow, it's amazing what cells can do. How are the organelles given to each daughter cell?
For mitochondria and chloroplast, there are so many of these in this cell that some always end up in each daughter cell by random chance. The ER remains in the center of the cell and cytokinesis cuts it in half. During M phase, the Golgi fragments into vesicles, and these vesicles attach to the spindle microtubules via motor proteins and hitch a ride to opposing cells.
That is very cool. What's next?
Next, we will talk about cell junctions. Epithelial sheets are clusters of epithelial cells that are organized. Epithelial cells are barrier cells. Epithelial sheets have an apical side that faces outward, away from the tissue, and a basal surface that faces inwards and rests on connective tissue. There are 5 different ways that epithelial cells connect to things, and they are all cell junctions. The first is tight junctions. Tight junctions seal cells together so that nothing can leak in between them. Claudines and occludines for tight junctions. They arrange like 2 sides of a zipper on either cell and zip up, sealing the 2 cells together. Tight junctions keep organisms alive and define the membrane domains. The next 3 are similar to each other. Adherens junctions and desmosomes bind epithelial cells to one another. Hemidesmosomes bind epithelial cells to the basal lamina. Adherens junctions and desmosomes connect cells using cadherin. Cadherins are tethered to actin filaments or keratin. Integrins and hemidesmosomes are linked to collagen. The 5th cell junction is gap junctions. Instead of being a seal or an anchor, these are openings. Matching connexons meet between 2 cells and create a continuous channel across the 2 cell membranes. Small inorganic and hydrophilic molecules can freely diffuse between cells connected by a gap junction.
I feel like I am learning so much. What's next?
Next, we discuss cancer.
Oh no.
Cancer is unique among other diseases because it's not caused by pathogens that invade you, it is you. Cancer is an example of things going catastrophically wrong in our bodies. Cells are social. They live in large multicellular societies with constant molecule and information transfer. Cancer is a violation of the cellular social order. Cells do what they do for the greater good of the organism. Cancer cells are selfish and asocial and do not do things for the greater good of the organism. A normal cell should divide when new cells of its type are needed and shouldn't divide when they aren't. They should undergo apoptosis once the need for them has passed. Cancer cells do not undergo apoptosis and divide even when they shouldn't be. If a cell that's selfish and doesn't undergo apoptosis enters this cell cycle, it will begin dividing and pass that asocial instability onto daughter cells. Cancer cells are defined by 2 main properties. They proliferate like crazy and they invade and colonize regions that are reserved for other cell types. Cells that have the first property, but not the second, become a benign tumor. If cells contain both properties, then the tumor is classified as malignant. The worst classification is metastatic. The tumor becomes metastatic when it has migrated or invaded a blood or lymph supply line. So malignant cells have gotten into the circulatory system. Once this happens, the blood or lymph carries the cells to other parts of the body. Then wherever the cell lands, it gives rise to secondary tumors. Epidemiology is the use of statistics when analyzing the relationship between certain environmental factors and human disease. Linking certain environmental factors to cancer could possibly remove or minimize our exposure to those factors. But cancer is not just a disease from the environment, it is a disease of cellular mutation. Cancer is unique as a genetic disease because it is exclusively a disease of somatic mutations. We are protected from developing cancer by an early age because of the randomness of the mutations. Cancer usually results from an accumulation of very specific mutations which come together in unique combinations. It is rare for these specific mutations to come together in the specific combination unless a sufficient amount of time has passed and a sufficient amount of combinations have occurred. That is why cancer is a disease of old age. Genetic instability is seen to be causally related to cancer progression. When mutations affect the accurate replication and maintenance of the genome, we see large-scale genetic instability and chromosomal mutations occur. Cancer cells survive better, overtake and outcompete healthy cells. Cancer cells no longer require signals from other cells to thrive, they do not default to apoptosis, they are immortal and never stop dividing, they are genetically unstable, have chromosome mutations, and are abnormally invasive. They can also adhere to, survive at and divide in foreign tissues of other cell types. There are two classes of gene mutations that could lead to cancer. Genes that when mutated and become hyperactive cause cancer are called oncogenes. The unmutated gene is called a proto-oncogene. The other is genes that when mutated and become inactive cause cancer, and they are called tumor suppressor genes. Tumor suppressor genes are genes that should be on to stop cancer from developing, but are mutated to become off. Oncogene mutations are genetically dominant, meaning you only need one mutated oncogene allele to develop cancer. Tumor suppressor genes are genetically recessive, meaning that you need two mutant alleles for cancer to develop.
Very interesting. Are there any treatments for cancer?
There is no one universal cure to cancer. The more we understand cancer, the better we can devise treatments. Early detection of a tumor means that it has had less time to mutate and can be more easily removed. Radiation treatment works by bombarding the tumor with so much DNA damage that the cells die while neighboring healthy cells can undo repair DNA damage. Cancer has limited to no DNA repair and no cell cycle checkpoints, so it cannot repair itself after radiation. There is a drug that works by inhibiting a more primitive DNA repair pathway that cancer cells do use to repair DNA. Our healthy cells do not use it, so it does not affect our cells. This drug combined with lower doses of radiation can eradicate a tumor. Some modern strategies involve blocking the formation of new blood vessels which would feed the tumor. Vaccines are also being developed that would allow our own immune system to recognize unique features on the cancer cell that causes the immune system to fight the cancer as it would fight an infection.
Wow. Thank you for teaching me so much.